All right, welcome back, everybody, to Bio 153 iNet Online. I'm broadcasting to you live today from my house. And so, broadcasting out to Delta College, Bio 153, throughout the Tri-Cities, the Thumb, and Northern Michigan. So, welcome back to all. So, we continue looking at Chapter 26, looking at the physiology portion. We've already looked at the anatomy portion in regards to understanding the parts of the nephron. Remember the nephron being the structural and functional unit of the kidney. So we'll go ahead and we'll dive right into our notes. So there's three important parts to the physiology, three important concepts to focus on in here today. So we'll go through that. First is the important concept of understanding what's known as glomerular filtration. Glomerular filtration. So this is a very important physiological concept, which ultimately is all about the formation of the filtrate, which we talked about a little bit last time. So what happens again, we form the filtrate, is you have blood that's in the glomerulus here. These would be the glomerular capillaries. And ultimately, items that are small enough are going to leave the glomerulus, go through the fenestrations, go past the podocytes, and will enter into the Bowman's capsule, or the glomerular capsule. And here we have the filtrate, which will continue on through the nephron itself. Or another way to look at it, right here again is the glomerulus. Items that are small enough will leave the glomerulus, make their way into this area here known as the Bowman's capsule or glomerular capsule. And ultimately we have the filtrate, which will continue on here and through the nephron itself. So that's a very important process, glomerular filtration. It's ultimately a measurement of how healthy your kidneys are based off that overall rate of forming filtrate. And any molecules that are greater than 5 nanometers in size will be able to form the filtrate. Of course, our exceptions to that rule would be the plasma proteins and red blood cells that are too big and bulky and will actually stay within the blood itself. And urine formation, as you can see down here, ultimately makes up less than 1% of total filtrate, which is typically waste products we have to get rid of. Another very important physiological concept that we're going to talk a lot about in here today is looking at what's known as tubular reabsorption, that process of tubular reabsorption. And so when we look at tubular reabsorption, we're ultimately taking items that are beneficial to the body, such as found in the nephron, maybe in the renal tubules, and ultimately these items being transferred from the nephron back into our blood. We want to hold on to these items. We can kind of see that here. Some of these items are listed. Nutrients are critical. We don't want to lose these. So glucose, amino acids, water, possibly hold on to some salt, maybe bicarbonate, that very important weak base, and so on and so forth. So again, when we look at tubular reabsorption, if this was the nephron right here's the nephron, ultimately tubular reabsorption, these items leave the nephron in yellow and go into our blood supply right here. In this case, the blood here going into these capillaries here, the tubular capillaries. So that's the idea and concept of tubular reabsorption. Materials are leaving the nephron and going into our blood. So this is a very important concept that is taking place in the nephron. The next really important physiological concept that we're going to be looking at in here today is the idea of tubular secretion. Tubular secretion. And this is getting rid of items that aren't beneficial to the body, that we want to eliminate and will go out through your urine. So things such as drugs, maybe it's alcohol, maybe it's marijuana, maybe it's cocaine or heroin or whatever it might be. Various toxins that could be coming about from the drugs, and even excess hydrogen ions and other things. So these items here are leaving the blood, so perhaps these items are in the tubular capillaries, and these items leave the blood and go into your nephron in the yellow here. So again, we want to get rid of these items, and ultimately they'll be in your urine. Hence, if someone was taking drugs and a drug test occurred, simply your analysis would be able to detect some of these drugs, alcohol that somebody may be taking on. Okay, so now our focus, we're going to really zoom in on understanding of glomerular filtration, this very critical process of forming filtrate. So glomerular filtration. So when we think of the direction of blood flow here, blood is going to be making its way down the afferent arteriole and eventually into the glomerulus, these network of capillaries here. The afferent arteriole typically is a high resistant blood vessel. It's under very high blood pressure, which drives the blood with materials into the glomerulus. But then it's a matter of getting these materials out of the glomerulus and into the Bowman's capsule, right? This area right here in the actual nephron. So there's going to be a variety of different types of pressure that's going to be critical. Overall, pressure is the driving force to drive materials out of the glomerulus, past the podocytes, and into the actual nephron, and then, of course, the filtrate moving on. So this picture is showing that. One of the big parts of the pressure here we're looking at is what's known as the glomerular hydrostatic pressure. The glomerular hydrostatic pressure. This is considered to be the chief force of pressure, which is pushing materials out of the nephron, like water and solutes, items that are small enough to, again, leave the glomerulus, going past the podocytes, and into the Bowman's capsule pictured here. And this is also represented, the glomerular hydrostatic pressure, by the purple arrows that you see here. Other types of pressure that are indicated here, we have the blood colloid osmotic pressure and the capsular hydrostatic pressure, indicated by the green arrows that you see and the light blue arrows. Those are considered, these two pressures, to be opposing pressures. They're going against the hydrostatic pressure. And the hydrostatic pressure is that driving pressure to, again, push materials out of the blood, out of the glomerulus, into the Bowman's capsule, forming filtrate. Now, you could put all of these pressure variables into this equation here, NFP, net filtration pressure of the capillary, and you may possibly get a reading of positive 10 millimeters of mercury. If you gain a positive reading, that's the indication we're getting materials actually into the nephron itself. And you don't need to worry about calculating this, but again, just be aware of these different pressures. Again, when it comes to forming the filtrate, materials that are less than 3 nanometers in size will form the filtrate. So here I have a list of various items here: water, glucose, amino acids, vitamins and lipids, so you see a lot of nutrients here. Various ions, salt. This is bicarbonate, that weak base we've mentioned several times in the past. And then even nitrogenous waste products like urea. Remember, urea is formed from protein metabolism? So they ultimately, these items form that filtrate. However, like I've mentioned though, there's two items that are too small, and these items, again, would be the plasma proteins and red blood cells, and they would actually continue on into this efferent arteriole right here, which is under low blood pressure. All right, a very critical thing to focus on for understanding, well, how efficient are my kidneys actually working? How well are they doing? Well, that's looking at what's known as the glomerular filtration rate, or known as the GFR. So the GFR is referring to the glomerular filtration rate, the volume of filtrate formed per minute in the kidneys. So this is a typical reading here, 120 to 125 milliliters per minute. So the GFR is directly proportional to total surface area, what type of surface area is available to form the filtrate, the filtration membrane permeability, and also the NFP, the net filtration pressure. So if someone has a lower GFR, it's possible then they may have some form of kidney failure, maybe chronic kidney failure or acute kidney failure. The number would be significantly lower, the GFR, if someone had acute. Acute, the kidneys are failing much quicker in comparison to chronic, where they could live quite a while with chronic kidney failure. Maybe they could live decades. So when it comes to answering these questions, what factor drives filtration? Ultimately, that answer is referring to the concept, again, of pressure. The concept of pressure. And then the bottom question here, damage to the filtration. If there is damage to the filtration available, it's possible that you could see blood in the kidney. You could see plasma proteins. And if you're seeing blood, we could be talking about some form of kidney injury, kidney trauma. So definitely not a good situation there for sure. Okay, the next thing we're going to take a look at is how kidneys play a role with changes in your blood pressure. And I know there's a lot happening on this slide, but we'll take it one bit at a time here, one step at a time. So ultimately, the regulation of glomerular filtration rate. Imagine you had a patient that their blood pressure is dropping, maybe because of shock or whatever it might be, blood loss. So here we have a change in blood pressure, and that's ultimately possibly going to lead to a change in your GFR. Your glomerular filtration rate may decline. And what will happen then with a decreased filtrate flow, maybe even salt decreasing, it actually will signal the macula densa cells we've mentioned last time, because they sense changes in salt. And ultimately, this could lead to vasodilation of afferent arterioles, and ultimately maybe leading to an increase in the GFR. Now, part of that could also be vasodilation of your afferent arterioles too. So again, macula densa cells playing a role here in regards to sensing changes in salt because of a fall in GFR. The granular cells, we mentioned those last time too. They play a big role in sensing changes in your blood pressure. So the case here, the effect of that with a fall in blood pressure is the release of the critical enzyme known as renin. And then renin will be important for the conversion of this important plasma protein here known as angiotensinogen, and the conversion of this to angiotensin II. Angiotensin II is a plasma protein here and very critical, acting as a vasoconstrictor. So vasoconstrictor, we mean vasoconstriction of blood vessels And that's very important, this idea of vasoconstriction, to help to get the blood pressure back to normal, the systemic blood pressure. Of course, there can also be the release of the hormone aldosterone from the adrenal cortex, which can increase salt reabsorption, leading to increase in blood volume, leading to increase in blood pressure too. So definitely take a look at this slide. I know there's a lot happening here, but make sure to definitely take a look at this and review it. What we're going to do now is go through the parts of the nephron together. So imagine that we have our filtrate. The filtrate went through the Bowman's capsule, and now we're into the proximal convoluted tubule. The proximal convoluted tubule. And this is where we really get to see the physiological processes of tubular reabsorption and tubular secretion working. So, important to know, the majority of the tubular reabsorption in the nephron takes place at the PCT, the proximal convoluted tubule. So there's a variety of items that are going to be reabsorbed here, items leaving the nephron and going back into the blood supply. That would be some water, so 65% of water is going to be reabsorbed here. You're going to have sodium reabsorbed here, ions, and even 100% of your nutrients like glucose and amino acids and lipids, vitamins, all reabsorbed here at the proximal convoluted tubule. You also have some tubular secretion occurring. Again, this is where we have items that are in the blood that move into the nephron, typically items that aren't beneficial to the body, that are harmful to the body. So things such as drugs, we got to get rid of drugs. Maybe too many hydrogen ions we got to get rid of, and possibly even ammonium. So NH4, so a nitrogen and 4 hydrogen groups. NH4 stands for ammonium. Ammonium. Imagine now that the filtrate has made its way now into the loop of Henle. And so there's going to be tubular reabsorption and some secretion here. Remember, we have 2 limbs to the loop of Henle, the descending limb and the ascending limb. First, we must go down before we go up. So imagine that the filtrate has entered into this descending limb of the loop of Henle first. And it's in this area of the loop of Henle, the descending limb, where you're going to have a lot of water leaving through osmosis. So a lot of water leaves here through the process of osmosis, which is a passive transport process. And very importantly, the filtrate will become increasingly concentrated with a high amount of solutes. So it's more concentrated because a lot of water is leaving the nephron, and the water is making its way into the surrounding tissues and can even be transferred to blood supply if need be. During the ascending limb, we go around the bottom of the loop of Henle and going up. The ascending limb is permeable to salt, sodium chloride. So a lot of salt is going to leave this section of the loop of Henle, and this is possible through secondary active transport. And as this is happening, as the salt is leaving, the filtrate now becomes increasingly diluted. And that's possible because of the salt loss, so a low amount of solutes now. The purpose of this movement of water and salt, obviously, is for overall balance of water and salt in your body to assist with homeostasis with proper levels of water and salt. So another important idea to look at in the kidney is this idea of the osmotic gradient that's found, especially in the renal medulla region. Remember last time we talked about 2 regions of the kidney, the renal cortex, this outermost layer, and then the renal medulla, the most interior, deeper region. And a very important concept to understand when it comes to water and salt loss in the kidney is this idea of osmolality. Osmolality. So this is kind of an important chemistry term here, osmolality. And so osmolality ultimately is looking at the number of solutes particles, so number of solutes in a kilogram of water. So really the ratio of the amount of solutes, things like salt, things like nutrients, etc., that we find in water. And osmolality, the units here, is what's known as milliosmoles. Milliosmoles. And the osmolality levels are lower up in the cortex area, but the overall amount of osmolality increases significantly as you go deeper and deeper into the medulla region of the kidney, and you get way to the bottom of the medulla. It might be around 1,200 milliliters of milliosmoles, I should say, for units. All right, so here we can see here the loop of Henle. Here's our loop of Henle here. And we can see these numbers correspond to the amount of osmolality, again in milliosmoles. So the level is pretty low up here in the glomerulus, Bowman's capsule, the PCT. But as the filtrate makes its way down through the loop of Henle descending limb, the number, when it comes to osmolality, is greater and greater as you're getting deeper and deeper into the innermost area of the renal medulla. Lots of water, you can see here, is going to be leaving during the loop of Henle in the descending limb. Water leaves through osmosis, and that water can be picked up ultimately in the blood, can be out in the surrounding cells. Water leaving through osmosis, and as the water leaves, once again, the filtrate becomes more and more concentrated all the time. The filtrate will continue around the bottom of the loop of Henle. The greatest amount here is 1,200 milliosmoles at the bottom of the loop of Henle here. So you have a lot of water that has left at this point. And now the filtrate continues up the ascending limb of the loop of Henle. And this is where you can see a lot of salt has left. A lot of that leaving through active transport, secondary active transport. And as the salt leaves, a lot of it, the filtrate becomes very, very diluted because of the loss of salt, that important solute. And then eventually, as the filtrate makes its way up here in the DCT, the distal convoluted tubulo, the numbers drop off significantly for osmolality. This happens a lot in the jugular medullary nephrons. They contain a very, very long loop of Henle, which allows for the production of concentrated urine, assisting with that. And again, the movement of the water and the salt ultimately is important for proper balance and homeostasis of water and salt in your body. One thing to consider too, the blood vessels that are going to be in close proximity to that loop of Henle in the jugular medullary nephrons is going to be the vasa recta capillaries here. So these are the vasa recta capillaries. They're important for maintaining the osmotic gradient when we consider movement of water and salt in the loop of Henle. They're delivering nutrient blood, so this is where you have actual movement of oxygen-rich blood taking place to tissues. So that's very important, the idea of gas exchange here. And the vasa recta capillaries protect the medullary osmotic gradient when it comes to movement of water and salt, so it's at a regulated level to have that proper balance in your body. All right, the next section of the nephron we're going to take a look at in here today is looking at the distal convoluted tubulo. The distal convoluted tubulo. So this is right after the ascending limb of the loop of Henle. This functions when it comes to tubular secretion and also reabsorption. In fact, the majority of tubular secretion takes place right here. So you have a lot of various items that we need to get rid of out of the blood into the nephron and ultimately out through urination. Here are all these items: organic acids, urea. So again, urea forms because of protein metabolism. You have a new material here known as creatine. So creatine, we got to get rid of that. That's kind of a byproduct from ultimately creatine. When we think of muscles, we have ammonium we got to get rid of, hydrogen ions, potassium, and drugs. And again, the drugs, we could be talking about a variety of things: alcohol, marijuana, cocaine, heroin, etc. All right, now, this is the first section of the nephron that is actually controlled by hormones, okay? This is the first section of the nephron that's actually controlled by hormones. So the hormones will have an influence here, especially when it comes to reabsorption of sodium, calcium. And reabsorption, these items leave the nephron and go into the blood. Reabsorption of sodium will be controlled by the critical hormone here known as aldosterone. So very important to know this hormone, aldosterone. While reabsorption of calcium will be controlled by the parathyroid hormone, or PTH. It's very possible you might have talked about this way back in the day in Anatomy 1 when you talked about bones and calcium. So make sure to know those two critical hormones, aldosterone and the parathyroid hormone, and what they control. Aldosterone will also control the movement of potassium here too, when we consider maybe there's too much potassium in the body, and hence the idea of tubular secretion. So aldosterone controls the reabsorption of sodium. Aldosterone controls the tubular secretion of potassium. The last part of the nephron we're going to take a look at in here today is the collecting duct. The collecting duct. And the thing to note about this collecting duct is that the collecting duct is receiving the filtrate from many, many nephrons. There's thousands of collecting ducts in the kidney, many nephrons ultimately draining their filtrate into the collecting duct. And like I've mentioned before, this is the first section where, for the first time, we can consider the filtrate is transformed actually into urine here, is at the collecting duct. Now, you're also going to have movement of materials here too. Hormones will be controlling the movement of these materials, so an influence with hormones here, just like the DCT. So when we think of items that are undergoing reabsorption, water, for example, water is going to be controlled by the hormone ADH, which stands for the antidiuretic hormone, ADH. So really important to know that one. And once again, sodium can be reabsorbed here in the collecting duct, controlled by the hormone known as aldosterone. And then also we don't want to forget that potassium, also controlled by the hormone aldosterone here when it comes to tubular secretion. Remember, we talked about reabsorption. Materials are going to be transferred from the nephron to the blood. We talk about secretion. Materials are transferred from the blood to the nephron to get rid of. So important to know these critical hormones here and what they control. All right, so it's important to have a good understanding of this hormone, ADH. And this is really key when we think of the regulation of water intake in the body. A lot of people don't drink enough water, and ultimately there's signals sent to your brain when it comes to this idea of this thirst mechanism. So when you think about this idea of water, does anyone know what part of the brain controls thirst? So take a moment to consider that. What part of the brain controls thirst? And hopefully you were thinking that the part of the brain that controls your thirst is the hypothalamus, right? The part of the brain that controls your thirst is the hypothalamus. So that's a good signal. Your mouth is dry, you got to drink water, and then you go ahead and drink some water, which I'm going to do right now, by the way. Oh, that water felt great. I tell you, nothing like having some water to feel just wonderful and alive. All right, moving on. So, we take a look again at the collecting ducts, and we look at the situation of how the important hormone ADH controls the amount of water in the body, plays a big role here, this antidiuretic hormone. So here we're looking at an example of someone that's dehydrated. So perhaps somebody wasn't drinking enough water. This happens a lot in the summertime. Maybe somebody's working outside for a long period of time, and they're working in their garden, they're cutting the grass, and they haven't been keeping up their water supply, and they're starting to feel dizzy, they're getting a headache, dry mouth. Those are all signals of some dehydration. So ultimately what will happen here is there'll be the release of the hormone ADH, and this hormone will be released, and you'll have an increase in ADH production. And the ultimate goal of ADH is to go ahead and increase water reabsorption here in the collecting duct. And so what will happen then is a decrease in urinary outflow. So when you're extremely dehydrated, you're probably not peeing a whole lot. Your urination is probably at a very low level. So that's kind of what happens when you're extremely dehydrated. Again, ADH is released, a significant amount of the hormone ADH, the antidiuretic hormone, and the effect of that then is a decrease in urinary output. We're now briefly going to talk about kidney stones. And kidney stones, a major cause why some people get them is dehydration. That's a major trigger, dehydration. Somebody's not drinking enough water. And so you can see in this X-ray image here, someone who has kidney stones, several here on the side here. So this is a blown up picture of a kidney stone right here. So imagine having one like this, 8 millimeters in length, extremely painful. It's very possible that maybe some of us have past kidney stones, but we didn't even know it because they're so small. But some people, they get kidney stones, dehydration is a major cause, and they actually can get stuck in the kidney, in the renal pelvis area, or even stuck in your ureter. And this can lead to excruciating pain. It may be so bad that eventually surgery is the only option, where ultimately there's surgeries involved, laser surgeries to pulverize the kidney stone, and then typically there's a stent that's put in place for a time being. However, anytime you have surgery, there's always a big chance, risk factor of infection. So sometimes when the stent is removed that was put up into the ureter kidney and you pull that out, sometimes infection can occur. So it's important to keep an eye out for that. But you can see the various items that make up the kidney stone. It could be lots of uric acids that form, magnesium, or even calcium. So you start wondering, well, how's my water supply? Some areas, some water supply has a lot of those various minerals. And literally that's something you can see around the U.S. in certain areas. It's probably better to get bottled water. It's something you can definitely see in Las Vegas, where the water coming out of the drain there's significant calcium, and the water coming out of the faucet is like a milky white fluid because of the calcium. And so it's really key not to drink that water because you could get kidney stones. Other causes for kidney stones other than dehydration, not drinking enough water. With that, of course, might be a high salt diet. So having a lot of foods that have high sodium could lead to kidney stone development, or even your genetics could play a role too. Once you get kidney stones, there's always a higher probability that you'll develop more later in life. So it's really important to alter that diet, low sodium diet, and drink lots and lots of water. All All right, diuretics. We've mentioned diuretics before, but here are some that can enhance urinary output. So things such as drinking alcohol, having a beer, drinking coffee with lots of caffeine in it, or even pop, tea, etc., can ultimately play a role with regulation of water intake, and ADH in some way or form might be triggered here. So here we're taking a look at the collecting duct here again. And in this situation, perhaps somebody's been drinking lots of beer, drinking Mountain Dew or Red Bull or whatever it might be. And when you're taking these items that are considered to be diuretics, that's going to actually suppress the hormone ADH. So a decreased amount of this hormone will be released. And because of that, you're not holding on to as much water in your body. And this leads to an increase in urinary output. So that's definitely the case with alcohol, caffeine intake for sure. So you've probably noticed that before if you go out to a brewery or go to a winery and you have a couple beers or a couple glasses of wine. You ever notice how the lineup to go to the bathroom is getting longer and longer all of a sudden? Well, ADH is being suppressed, this hormone, and that leads to lots of urination that's going to take place. And it could lead you to becoming dehydrated eventually too, by the way. Other things that could lead to suppressing of ADH would be elevated water intake, someone who drinks way too much water, or diuretic drugs. So various cardiac patients that are taking diuretic drugs, taking Lasix, for example, could lead to increased urinary output. All right, so we're going to take a moment to review this idea of tubular reabsorption and secretion. So I'm going to go through this together with you for review here. Maybe you could do this at home. Maybe you could go ahead and draw a nephron. That's what this is again, a nephron. All right, so this first part here we're going to look at again. This is the proximal convoluted tubulo. So the PCT here. So PCT. So our proximal convoluted tubulo right here. The PCT. And first we'll look at, well, what types of items are going to be reabsorbed? In other words, what items are going to leave the nephron and go back into the blood supply? Ultimately here, you're going to have all of your nutrients. So 100% of your nutrients are going to be reabsorbed at the PCT. So again, that's referring to glucose, amino acids, vitamins, and lipids that are reabsorbed. You also can have some water reabsorbed here. So keep that in mind. Water, H2O, about 65%. Likewise, you can also have some ions reabsorbed here. So ions. Might be kind of hard to read there, but IONS, ions. And then those would be the items that are typically reabsorbed. Oh, don't want to forget, I thought I was forgetting one here. Yes, sodium. So we don't want to forget our sodium, yes. So you also have some sodium reabsorbed here too. So these items are reabsorbed. But you might be asking yourselves, well, Chris, what items are going to be undergoing the actual tubular secretion here at the PCT? So there are some items here. So items such as drugs. Again, drugs, we're talking alcohol, we're talking marijuana, we're talking heroin, things like that. So drugs undergo tubular secretion at the PCT. Maybe excess hydrogen ions, perhaps ammonium, so N H4 plus ammonium. So those are some great examples of items that undergo the tubular secretion versus tubular reabsorption. All right, moving down here. Here we're looking at the loop of Henle. So first taking a look at the descending limb. So we'll start over there. Taking a look at our descending limb, because first you must go down before you can go up. So taking a look at the descending limb. So descending limb. The main item that is undergoing tubular reabsorption here in the descending limb, of course, is going to be your water. Water leaves through osmosis in the descending limb. And so the filtrate, again, becomes very concentrated with the water leaving, especially when you get to the bottom of the loop of Henle. And then when we go up here, we go up. Now we're taking a look at the ascending limb. So now we're taking a look at the ascending limb of the loop of Henle. The main item leaving here through reabsorption will be your salt, sodium chloride. The sodium chloride is leaving through secondary active transport. So that's the main item leaving here. So as the salt leaves, the filtrate ultimately becomes very diluted with the loss of that major solute salt. All right, up here, this twisted coiled section here, this would be your DCT. So the distal convoluted tubulo up here. So when we take a look at this, this is the major location for tubular secretion. Remember, the PCT was the major location for tubular reabsorption. DCT, major location for tubular secretion. So the variety of items that undergo the tubular secretion, I'm going to write them over here because we're running out of room here. These items would be such as drugs. So again, alcohol, marijuana, cocaine, heroin. You also have ammonium again leaving here. So NH4 plus. You also have hydrogen ions leaving here. You also have potassium leaving through secretion here. Urea, byproduct of protein metabolism, as a great example here. Organic acids. So organic acids. So a lot of different items are leaving here. Also creatine, but I kind of ran out of room here. I guess I could write that down here for you though. So creatine. So CREATINE. Typically formed from coming from muscles, some breakdown of muscle. Okay, so those items leave through tubular secretion. What about reabsorption, right? Well, the one major item when we think of reabsorption here, two actually I should say, would be Sodium is reabsorbed in the DCT. So sodium and calcium, Ca plus 2. All right, and then finally, this is the collecting duct, right? So many things undergo tubular secretion. The DCT, I kind of ran out of room here, but this yellow vertical structure going down is the collecting duct. So the collecting duct. And items that are going to be reabsorbed here that we're going to hold on to, that would be items such as water in the collecting duct. So water. Likewise, sodium. So those are items that undergo reabsorption. And then for tubular secretion here, the one item here in the collecting duct would be potassium that undergoes the tubular secretion. I noticed up here with the DCT, again, all these items over here undergo tubular secretion from blood to the nephron. And for these two items here, again, for reabsorption, it's going to be that calcium and sodium leaving the nephron going to the blood, just to go ahead and clarify that. Now, there's also going to be some hormone influence, and so we'll erase our board here. Maybe you're going to erase yours at home. And I want to talk about that. So, the two major locations where hormones have an influence on the nephron is your DCT and your collecting duct. So DCT, distal convoluted tubulo, and the collecting duct. All right, so what hormones are we talking about? Well, let's go ahead and take a look. So, when we talk about water, H2O, water is going to be controlled by ADH. ADH, this hormone, refers to the antidiuretic hormone, and it only plays a role in the collecting duct. So, especially with water reabsorption. So, an arrow going out like this. Water going out, controlled by ADH. All right, next, calcium. Calcium is controlled by the hormone known as PTH. PTH. PTH stands for the parathyroid hormone. Parathyroid hormone. And this reabsorption is taking place in the DCT. So I have an arrow going out like this here for the calcium, leaving the DCT and going to blood. Sodium. Sodium is going to be controlled by the hormone known as aldosterone. Okay, so that's really important to remember. Aldosterone. So reabsorption of aldosterone is extremely important when we consider the DCT. and also when we consider the collecting duct. And then finally, we don't want to forget about our potassium. Potassium is also controlled by the hormone aldosterone when it comes to tubular secretion. So, in other words, potassium coming back into the nephron from the blood, controlled by aldosterone, tubular secretion. And likewise, potassium being controlled when it comes to tubular secretion in the DCT here too. So, very important to make sure to review the two sections of the nephron controlled by hormones, the DCT and the collecting duct, and knowing that water, calcium, sodium, and potassium are controlled by hormones. Water through ADH, calcium through PTH, and sodium and potassium controlled by aldosterone. So definitely make sure to review this. Very critical to ultimately know this. So please make sure to review this, because you could see this on definitely an exam for sure. So definitely make sure to review this. Okay? All right, next, taking a look now at renal disease, renal failure. So there's the acute condition and chronic. Remember, this is all based off the GFR, and you can get a GFR reading based off of a blood sample. So someone's considered to have chronic renal failure if your GFR is less than 60 milliliters per minute. Chronic, more long-term, you can live with it longer. Acute renal disease, a GFR rating less than 15 milliliters per minute. So acute, that's a pretty serious condition. Your kidneys are barely operating. Major causes for this would be things such as someone developing a severe kidney infection, perhaps from kidney stones, someone who's an alcoholic or taking lots of drugs, a drug abuser, maybe they're addicted to heroin or something. Physical trauma, so injury to the kidneys, someone that has diabetes, maybe type 2, type 1 definitely can lead to kidney disease. High blood pressure, all these items can lead to kidney failure. When it comes to treatment, maybe at first they're using hemodialysis, but eventually that may not be effective. And so the only alternative is to have a kidney transplant. So this is a picture here showing hemodialysis. So a patient would be going in, maybe they're going through monthly hemodialysis, weekly hemodialysis, it just depends on their condition. And ultimately this machine acts as an artificial kidney because their kidneys aren't operating like they should. And so this machine will actually go ahead and filter the blood of waste products and excessive fluid. Ultimately, it may come down to a kidney transplant. This is just some statistics for someone that may have a kidney transplant. So 90% of kidneys donated by a close relative. The kidney can still function 2 years after the transplant. And 75% of kidneys donated from unrelated individuals can still function 2 years after the transplant. Of course, it's a matter of the body accepting the organ, that there's no organ rejection, and blood typing matching, that's important too. How this works, typically you'll have the donated kidney is usually inserted where the pelvis would be. You already have the ureter from the previous person, and that ureter then is attached down close to the individual's bladder. The renal artery and renal vein would still be attached from that donor kidney, and they're typically attached ultimately farther down, very close to where that common iliac artery, common iliac vein would be, typically speaking. And in most cases, the kidney that's not working properly anymore, sometimes that's just left in the person's body. And it can just lead to tremendous complications in surgery if they try to remove that, so it ultimately just doesn't function anymore. We now take a look at the bladder and urethra. So imagine we have that urine, it went down the ureter into the bladder now. And the area where the urine will collect first is known as the trigone. The trigone. Of course, this triangular area, unfortunately, this area is prone to UTIs. And then ultimately, the urine goes out through the urethra. There are two sphincters involved that help with urination: the internal urethral sphincter and the external urethral sphincter. The internal urethral sphincter is found in the smooth muscle lining of the urethra and involuntary. So we have no conscious control. Simply the sphincter will go ahead and relax. The external urethral sphincter is found in skeletal muscle, more outside, and is voluntary. So we do have conscious control over that sphincter. And so that's the sphincter we relax when we go to the bathroom and urinate. This is just a picture now more of the male urethra, but ultimately kind of the same idea here. So you can definitely take a look at this. And the last physiological concept we're going to take a look at in here today is looking at the concept of urination, or the fancy term for this, the process of micturition. So micturition, urination, the act of urinating. Of course, at first, we have no conscious control over urination. We don't really develop that till around approximately age 3, because the sphincters need to form. So early on, it's important for us to have our diapers. We need to go through potty training. Statistically, usually young girls are potty trained quicker than young boys. This picture here ultimately is looking at the physiology involved with the process of urination, or also known as micturition. So imagine you're drinking lots of water, or maybe lots of punch or whatever, and you're ultimately then filling up that bladder. And the bladder walls are stretching. This eventually leads to impulses being sent up to the brain, up to an area known as the pontine control centers. Again, they undergo maturation during the first couple years of our life. Signals then are sent through the pontine micturition center, eventually down to the spinal cord, and this will eventually lead to an increase in parasympathetic activity and a suppression of sympathetic activity. In turn, this will lead to the detrusor muscles along the lining of the bladder to contract. The internal urethral sphincter opens, so those signals sent back to the brain ultimately decrease in somatic motor nerve activity, and the external urethral sphincter opens. That's ultimately up to you, and then you pee. Eventually, when parasympathetic activity decreases, sympathetic activity will increase, suppressing the process of urination. Ultimately, this entire process, you can see here, is controlled by the parasympathetic nervous system, this process of urination, or also known as micturition. All right, so now we have some review questions here to look at. So, time to do your eye clickers. Find those eye clickers. So number 1, during glomerular filtration, what items are too big based on molecular size and stay in the bloodstream? So what would be the correct answer here? What would be the correct answer here? Okay, and the correct answer here should be choice letter B, both A and B. Plasma proteins and erythrocytes, red blood cells, stay in the bloodstream. Number 2, the majority of tubular reabsorption 100% nutrients occurs at what section of the nephron? So what would be the correct answer here? What would be the correct answer here? Okay, and the correct answer here would be choice letter C, the proximal convoluted tubule. Number 3, which of the following items undergo tubular secretion in the distal convoluted tubule? All right, and the correct answer here would be choice letter E. Both A, B, and C would be correct. All right, number 4, water is the only item of the filtrate that leaves this section of the nephron through osmosis called the what? So what would be the correct answer here? What would be the correct answer here? And the correct answer would be choice letter B, the loop of Henle descending limb. Number 5, which sections of the nephron are controlled by hormones? Which sections of the nephron are controlled by hormones? All right, and the correct answer here would be choice letter E. Both C and D would be correct. Both C and D would be correct. The distal convoluted tubule and the collecting duct. So very good. And number 6, you decide to take a road trip to Bell's Brewery in Kalamazoo. After having several beers, this will lead to blank and blank. So what would be the correct answer here? All right, and the correct answer here would be choice letter C. Decrease ADH levels and increase urinary output. ADH will be suppressed because of drinking the beer, okay? Which unfortunately is a diuretic and you'll pee a lot. So I hope you enjoyed today's lecture. This is the physiology. Of course, if you have questions, let me know. And I hope you have a wonderful day.